Now, when we ask a question in science, there are a series of steps that we need to go through so that it's systematic and logical. And we call this the scientific method. There are many different treatments of the scientific method, but these are the 5 basic steps we're going to use: observation, hypothesis, experiment, data, and analysis. So let's go through each step. Our first step in the scientific method is very simply making an observation. Hey, that's a neat thing that I saw and I'm curious about that. I wonder, does this work or how does this work? Observations generally lead to further questions and to making predictions. So let's say, for instance, that you're curious about the effects of fertilizer on a plant. So maybe what you want to do is get a nice little house plant like this, that's a plant, and put some fertilizer on it and see what happens. Your observation is that fertilizers might make plants grow faster. The second step of the scientific method, and actually the most important step, is called the hypothesis. A hypothesis is a tentative explanation for why your observation is occurring. It's an educated guess. And this guess actually links together two of the variables, two of the things you observed. There is a proper format for writing a hypothesis, and I would like you to use this format in this class. And essentially you say, well, if A is true, then B must occur because of some reason. So if I see A happening, or if I change A, then B is going to happen. And because would be why I think that happens. A hypothesis is a claim that there is a relationship between two variables, two things in the situation. And these variables are known as the independent variable and the dependent variable. Let me give you an example to explain what these things are. The independent variable is the variable being tested. So let's say you're testing that fertilizer thing on your plant. So the effect of fertilizer is what you want to test. That's your independent variable. Okay? Now the dependent variable is the thing that will change in response, or the thing you're going to measure. So let's say that in this experiment you're going to measure the height of the plant. That is going to be your dependent variable. Now you can use these two variables to make your hypothesis. You can say, if I put fertilizer on the plant, then it will grow faster. And you could add a because, something about nitrates or nutrients in plants, if you wanted to add a because. But a good hypothesis says, if I change the independent variable, then the dependent variable will change in a certain way because some reason. So there's a relationship between these two variables. After you've written your hypothesis, you can perform an experiment. An experiment is the actual testing that goes on. And the design of an experiment is really important. Good experiments minimize variables. Now, what do I mean by a variable? Well, a variable is any factor that could affect the outcome of your experiment. So you're testing your independent and your dependent variables, but there are always going to be others involved too. For instance, with a plant experiment, you can have all of these other variables going on. You can have predators such as slugs nibbling on your plant, the wind, the amount of sunlight, the temperature and the pressure, the amount of water that you're giving it, the type of fertilizer you're giving it. All of these things, even the soil, can affect how that plant grows. In order to minimize these variables, you want to keep these factors as constant as possible. And if you can't control the variable, then you really need to record that variable. You need to keep measurements of it and see how it changes. That way you can kind of account for it in the results of your experiment. So good experiments try to minimize variables. An excellent way to help minimize variables is to run something known as a controlled experiment. A controlled experiment involves replications of your original experiment. But in these replications and these other experiments, the independent variable has not been changed. So they represent kind of the natural or normal state before you started changing stuff. Controlled experiments are important because this control group gives you something to compare your experiment to. It helps validate that relationship that you are claiming exists between your independent and dependent variables. Let me give you an example. Here we have an example of a controlled experiment. Let's say you're doing your little test with the plants and the fertilizer. You have a plant here, you've been putting fertilizer on it, and you've been measuring the growth of the plant over time. Now your plant is growing, and that's awesome, it looks really good, but you can't really say that the growth is due to the fertilizer if you don't have anything to compare it to, because you don't know how the plant was going to react normally without the fertilizer. So what you might do is include a control. So here's our experimental group, right here. Here's our control group. So here we have a plant that's the same type, maybe the same size, same health levels, and you are treating it with just water instead of water and fertilizer. Now the variables between these two plants needs to be the same. So the atmospheric pressure, the amount of sunlight, the exposure to predators, all of that stuff needs to be the same between these two groups so you can make an accurate comparison. The only thing that you want to differ between these two groups is the fact that this one gets fertilizer and the fact that this one doesn't. That way, if the fertilizer plant grows faster than the water-only plant, you can say that it's the fertilizer causing the increased growth. Now, this particular type of control is known as a positive control. It's known to produce certain results, and positive controls usually sort of represent the natural conditions before you started changing things. Another type of control that is sometimes used is known as a negative control. In a negative control situation, typically something is removed so that you get the opposite results to the positive control. Negative controls are not always necessary, it just depends on the experiment that you want to do. But in this case, if we wanted to add a negative control, maybe we could have a plant that we gave no water to. Maybe we suspect that the water is making it grow more than the fertilizer, so we'll give this guy no water and see what happens, and there's my prediction about what it will probably look like. That's a negative control. As you perform your experiment, you're going to be collecting data. And data can be presented in a number of different ways. It can be quantifiable or qualifiable. It can be presented in tables or charts or pie graphs. It can be mathematical proofs. Here we have graphs, different kinds of graphs. It can be photographic evidence. It can be really nice drawings or even really good descriptions of what you see going on. In this next step, you're going to analyze your data and you're going to draw conclusions about what you're seeing. This is the point at which you are going to relate your hypothesis back to your data. And you're going to say, okay, does my data support my hypothesis or not? Does it make logical sense? Was I able to repeat the experiment more than one time and get the same results? Is my data reliable? This is the point at which you're going to decide whether to accept or reject your hypothesis. We really say accept and reject in science rather than prove and disprove, because prove makes it sound like you're at the end of the process, and really you're not. Now remember that rejecting a hypothesis is not a bad thing, because science is a process of elimination. But this is the point at which you will make that determination. You might decide that you need to go back and tweak your hypothesis or tweak your experiment and keep going, or you might decide that you've arrived at the answer you were looking for. Here's the data I collected from our little plant experiment. Kept track of the height over a month, and you can see the growth when we began, without fertilizer and with, and at the end. And very simply, you can see that the plant with fertilizer grew faster than the plant without fertilizer. So from this, we might conclude that fertilizer increases plant growth. We might accept our hypothesis. Again, we might want to go back and tweak our experiment and change it a little and try it again. Maybe more fertilizer makes it grow even faster. You might want to test that. Okay, but that's the analysis and conclusion part of the process. Now in this class, usually you'll just run an experiment once, and that's sufficient for our needs. But in real science, at this point in the method, we're going to start over again. Again, we're going to tweak that hypothesis, tweak the experiment, and try to replicate our results and do these experiments over and over again. If we were doing a plant fertilizer experiment, we wouldn't just have 2 or 3 plants. We'd want hundreds of plants so that we could really collect a lot of data and make sure that variables like genetic variation among the plants wasn't affecting how they grow. So this is just the beginning of the process all over again. Time for another year turn. You'll find this year turn in the guided notes. In this year turn, what I want you to do is think about what might happen if you go out in the parking lot and your car won't start. You can actually use the scientific method to try and figure out what's wrong with your car. And so what I want you to do is brainstorm that and work through the steps. Your observation is that your car won't start. What possible hypotheses could you come up with for why the car won't start? What experiments would you do to test this? What data would you collect? And what conclusion would you draw depending on what data you were able to collect? Okay, write this out and bring it to class with you.